Hello, everyone, and welcome to today's live broadcast, Development of Anti-Cancer Gene Therapies Through Understanding of Cytokine-Induced Programmed Cell Death, presented by Leah Persaud, a PhD candidate at Lehman College in City University of New York, the Graduate Center in City University of New York. I'm Susie Valdez, and I will be your moderator for this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. To learn more about our sponsor, Thermo Fisher Scientific, visit their website at thermofisher.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on that green Q&A button located at the lower left of your presentation window, type the questions into the box that appear on the screen, and we'll answer as many questions as we have time for at the end of the presentation. For questions not answered today, we will follow up with our participants via email. Also, please notice that you will be viewing the presentation in the slide window. To enlarge that window, just click on the icon located on the lower right of the screen. If you have any trouble seeing or hearing this presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know you're having a problem. Please join me now in welcoming Leah Persaud. Leah is passionate about understanding the molecular signaling pathways of new anti-cancer therapeutics. She is a City University of New York science scholar working in the lab with Dr. Moira Sauane's Cancer Research Lab at Lehman College in the Bronx, New York. She is a PhD candidate from CUNY Graduate Center. Leah has completed her undergraduate degrees in biology and forensic science from the University of New Haven in Connecticut. As an undergraduate, she performed research on highly specific DNA nucleases for gene therapy at Justus Liebig University in Giessen, Germany, which ignited her interest in molecular biology research and therapeutics. As a mentor, she's able to share her passion for science research and encourage undergraduate and high school students in pursuing their careers in STEM fields. Join me now in introducing and welcoming Leah Persaud. Welcome, Leah.
Hello, everyone. Thank you, Susie, for that introduction, and thank you, everyone, for signing in today. I'm Leah Persaud, a fourth-year PhD student studying molecular cell and developmental bio at the City University of New York and the Graduate Center in New York City. As Susie mentioned, I work with Dr. Moira Sauane at Lehman College, one of CUNY's senior colleges in the Bronx, and this is where I perform the majority of my cancer biology research. Today I'm excited to talk to you all about how our lab is tackling cancer by developing anti-cancer therapeutics for gene therapy. So let's get started. Okay, so as many of us know, cancer is one of the leading causes of death worldwide, right below heart disease. Over the years, the rate of death due to heart disease has slowly declined, but unfortunately, we do not see any declines for the rate of deaths due to cancer. To control cancer, prevention and early screening is key. According to the World Health Organization, 30 to 50% of cancers could be prevented by reducing risk factors such as alcohol and tobacco consumption, unhealthy diet, and getting vaccinated against cancer-causing agents such as HPV, also known as human papillomavirus, or hepatitis B or C viruses, to name a few examples. Despite this, however, cancer can also be genetic and even arise spontaneously, and it can go undetected if proper screening is not utilized. Once an individual forms a malignant tumor consisting of abnormally fast-growing cells, treatment is necessary to extend the life of the patient, to preserve quality of life, and also to reduce suffering. So currently, there are around five standard treatment options for individuals who have various stages of cancers. Generally, when cancer is first detected, surgery and radiation are used to physically remove or destroy cancer cells at a specific location in the body. When cancers have advanced, they evade the body's immune system, they become more aggressive, and thus they can spread to other organs. Thus, more specialized treatments are needed to attack rapidly proliferating cancer cells. Some of these include hormone therapy, chemotherapy, targeting therapy, such as gene therapy and personalized therapy. So gene therapy is very interesting because of its personalized approach. When developing any treatment plans for cancer patients, it's very important to look at the patient's genetic history and the mutational status of the patient's tumor. Not all tumors are alike genetically, and as a cancer advances, even tumor cells can differentiate and become less sensitive to treatments that were once effective in said patient. Because of this, no one treatment fits all cancer patients. Depending on the composition of a patient's tumor, different therapies need to be given to different patients. Essentially, the goal of gene therapy in cancer is to introduce functional genetic material into human cells to be transcribed and translated in order to regulate, repair, or suppress a molecular mechanism that contributes to cancer growth. By understanding the composition of a patient's tumor, the goal is to only target cancer cells while leaving normal healthy cells alone. Thus, gene therapy goes hand in hand with personalized therapy. So of all gene therapy clinical trials worldwide, over 64% of trials are for cancer treatment, for cancer diseases. Because of the ever-changing nature of cancer, gene therapy is becoming a more popular approach. So essentially, instead of having a one-size-fits-all or most approach, the goal is to create tailor-made therapies for patients who have tumors with different genetic profiles. And just recently, the Food and Drug Administration announced the approval of ex vivo chimeric antigen receptor gene therapy, also called CAR-T therapy, which will be the first gene therapy provided to the U.S. market to treat leukemia. The therapy was developed by Swiss pharmaceutical company Novartis, and basically it's a form of ex vivo gene therapy where patient T cells are removed and engineered to express receptors that will bind to patient cancer cells and then kill them once they are infused back into the patient. The therapy was approved to treat pediatric and young adult patients with leukemia, and this is really exciting news for the personalized treatment of cancer, and we hope to see additional gene therapies approval in the coming years. So, in addition to ex vivo treatments, there are in vivo treatments, where genes are directly put into a patient. Genes being developed for cancer therapy code for a variety of proteins, including tumor suppressors, specific antigens, transcription factors, cell cycle regulators, receptors, and cytokines. Our lab specifically studies a special cytokine called interleukin-24, as abbreviated IL-24. Interleukin-24 is of special interest in gene therapy because it has a selective killing effect on numerous cancer cell types while having no effect on corresponding healthy cells. So to give some background, interleukin-24 is a secreted cytokine, and it's a member of the interleukin-10 cytokine family. It was first discovered in melanoma cells and initially named MDA7, which is melanoma differentiation associated gene 7. Interleukin-24 is normally produced by immune cells and epithelial cells, and under physiological levels, it has various roles in autoimmunity, psoriasis, and wound repair. For the past 15 years, however, interleukin has been studied as an anti-cancer molecule because it specifically targets cancer cells and not normal cells. This is the really interesting thing about interleukin-24 because when it is overexpressed in cancer cells, it kills a variety of cancer cells from melanoma to glioma, prostate, breast cancer, and including breast cancer stem cells. Both overexpression in viral vectors and administration of recombinant interleukin-24 protein have been effective in inducing apoptosis in multiple cancer cell types, including cancers in transgenic mice studies. Interleukin-24 can also cause a bystander effect in which untreated cancer cells adjacent to treated cancer cells with interleukin-24 will undergo apoptosis as well. This is because it can induce its own expression of endogenous interleukin-24. So therefore, it does regulate its own expression. The most important thing about interleukin-24 is that it does not kill normal cells, and this has been confirmed in many studies. So because of interleukin-24 specific cancer-specific killing effect, it has been studied in phase 1 clinical trials, and it is currently in phase 2 clinical trials to determine efficacy and evaluate safety. So as many of you know, before a drug can be put on the market, it has to be tested in a series of clinical trials and be approved by the FDA. Before any of this can happen, the drug must be understood on a cellular level. So currently, interleukin-24 is being studied at phase 2 clinical trials. However, these trials have been limited to patients with only melanoma. To maximize the effect of interleukin-24 to different cancer cell types, the goal of our lab is to fully understand how interleukin-24 selectively kills cancer cells. We want to know its mechanism of action and what proteins it's interacting with to induce apoptosis, also called cell program death. Basically, we want to know what molecular mechanisms are being changed due to interleukin-24's overexpression. The way we do this is by performing basic research using cell lines and protein analysis. So in our labs, in order to do this type of preliminary pathway research, we use cancer cell lines as our model. In general, cancer cell lines replicate quickly, they are easy to take care of, and they are inexpensive compared to working with live animals. Also, there are a lot of data on cell lines because they are used in many labs across the world. So here we have an image, a microscopic image of one of the most routinely used breast cancer cell lines called MCF7. This cell line originated from a 69-year-old Caucasian woman who had cancer of the human mammary gland in the breast. These cells could be cultured in an artificial environment and maintained for long periods of time, usually longer than primary cells, which come from human or animal tissue, but they have a limited lifespan. These MCF7 cells are called immortalized cell lines. So cell lines are essential for us to study the mechanism of action of anti-cancer therapies, including interleukin-24. We can then plate cells in a variety of dishes for a variety of assays, such as those that measure cell apoptosis, cell proliferation, mitochondrial function, or reactive oxygen species production, also called ROS. In our lab, we treat our cancer cells with an adenovirus that contains the interleukin-24 gene. This same virus has been used in clinical trials and is non-replicative. We can treat cells with various protein inhibitors and determine if those proteins are essential for interleukin-24's killing activity. We can also use cell lines that have proteins that are mutated to determine if they are needed for interleukin-24 mediated apoptosis. We can also do really cool immunofluorescent experiments to determine if interleukin-24 expression translocates essential proteins to different parts of the cell, which will give us an indication of their function in response to interleukin-24 expression. So despite all of these advantages that cell lines offer us, they do have several limitations. Our cancer therapy research needs to be progressed with translational studies first through preclinical animal studies and then clinical trials. This, of course, takes time, but it is necessary to learn how therapies work in a living in vivo system. As many of my fellow researchers may also know, contamination is also a major issue. Contamination of cell lines, whether it be the introduction of microbes such as yeast cells, which you can see here, or cross-contamination with other cell lines, it can severely delay our experiments. In addition, if cell lines, especially cancer cell lines, are grown for too long, they can start to accumulate genetic mutations and exhibit abnormal morphologies or altered functions. This can affect the reliability of our results. Despite these disadvantages, we find cell lines extremely useful for our type of research, which is the understanding of interleukin-24's mechanism of action on a molecular level. It is important to test your hypothesis in vitro first and then progress to animal studies to save time and also effort and also money. Thus, overall, before any drug can be made for market, it must be discovered and tested in cell lines. So back to our protein of interest, interleukin-24. Thus far, this is what we know about interleukin-24. Interleukin-24 inhibits metastasis and also angiogenesis, and it induces apoptosis in many cancer cell types. It is able to inhibit metastasis by upregulating and downregulating factors involved in cell adhesion and migration. It also inhibits angiogenesis by downregulating angiogenic factors such as VEGF, FGF, and TGF. These are various growth factors. Most importantly, interleukin-24 activates apoptosis or programmed cell death by endoplasmic reticulum stress, also called ER stress. Interleukin-24's effect on ER stress in cancer cells has been well studied, and it is believed that this is one of the main mechanisms in which interleukin-24 induces cell death. So in general, cancer cells are in a constant state of ER stress. The ER is extremely important for cancer cells to thrive because this is the primary site for protein synthesis, which is essential for the rapid proliferation of cancer cells. And because cancer cells rely heavily on protein synthesis, there is an accumulation of misfolded mutant proteins, which causes a constant state of ER stress. With a higher ratio of survival and proliferation signals to death signals, the cancer cells can adapt to this condition and survive. In terms of interleukin-24 action, we and others have shown that interleukin-24 is potentiating ER stress to such an extent that cancer cells cannot recover. Death signals are amplified, and the cells thus undergo apoptosis. More specifically, interleukin-24 induces a buildup of cytosolic calcium, ceramides, and reactive oxygen species, which can lead to an apoptotic response. Our lab has also discovered that interleukin-24 interacts with a ligand-mediated receptor called the sigma-1 receptor in the ER. This interaction antagonizes the sigma-1 receptor, and it is necessary for interleukin-24 to induce its apoptotic effect. Oftentimes, in cancer cells, sigma-1 expression is upregulated. In our experiments, when sigma-1 receptor is overexpressed by adenovirus vector, interleukin-24's killing activity is actually decreased. So, to further understand how interleukin-24 induces apoptosis through ER stress, we have to understand what happens in response to ER stress. So in general, the cell will activate 3 different pathways to restore cellular homeostasis by increasing folding capacity, protein degradation, or by decreasing translation or protein synthesis. So these pathways include IRE1, PERK pathway, and the ATF6 pathway. So as I just mentioned, extreme and prolonged stress conditions of the ER can contribute to the induction of apoptosis. So we believe that interleukin-24 is doing this through the inhibition of protein synthesis by activating the PERK pathway, which is responsible for translation attenuation, so for the decrease of protein synthesis. We and others have shown that interleukin-24 does activate PERK to induce apoptosis. So one of our main objectives, one of our main projects was to determine if interleukin-24 specifically blocks protein synthesis when it is overexpressed in cancer cells. So the reason why this would be promising is because therapeutics that attack translation are essential to killing cancer cells because they can disable the machinery necessary for cancer cells to grow and proliferate. Pursuing translation inhibition can also be a more targeted and effective approach that can be used against a variety of cancer cell types that exploit different mutated pathways based off of their genetic profiles. So these therapies could be used against tumors that are heterogenic, in which the subsets of cells in the same tumor can have varied morphological, genetic, or metabolic, or metastatic characteristics. And since interleukin-24 exhibits a broad anti-cancer effect in many tumor cell types, its killing effect could be attributed to the inhibition of translation as a downstream pathway of ER stress. So more specifically, we wanted to look at translation initiation, which is the first step of protein synthesis. To initiate translation, the ternary complex must be created. This complex contains the methionine tRNA, GTP, and eukaryotic initiation factors to begin translation at the start codon. So a key eukaryotic initiation factor is called eIF2 alpha. Phosphorylation of eIF2 alpha on serine 51 specifically can block translation initiation, which is the rate limiting step in protein synthesis. We know from previous experiments that interleukin 24 activates ER stress and phosphorylates eIF2 alpha in lung and prostate cancer cells. So also, in addition, interleukin 24 is able to increase the expression of downstream markers of the ternary complex. So our goal here was to definitively determine if the anti-proliferative effect of interleukin 24 is mediated by phosphorylation of eIF2 alpha and ternary complex depletion in different cancer cell types. So thus, we want to determine if interleukin 24 is an inhibitor of translation initiation through this main mechanism. So one of the first things we did was to measure eIF2 alpha phosphorylation in a variety of cancer cell types, such as melanoma, breast cancer, cervical cancer, and squamous cell carcinoma. The way we would do this is first we would grow our cancer cells, and then we would treat our cells with the interleukin 24 gene in adenovirus. The cells would uptake the adenovirus within 2 hours, and then we could perform assays on the cells within 24 to 72 hours, depending on the type of assay we wanted to do. So some of these downstream assays include Western blot. So Western blot is for protein analysis to determine if specific proteins are affected by interleukin 24 expression. We can also do cell viability assays or cell proliferation assays that measure whether the cells are living after interleukin 24 treatment. We can also do annexin V apoptosis assays that measure how many cells are undergoing apoptosis using flow cytometry. And we can also do qRT-PCR to determine what genes are getting expressed in response to interleukin 24 treatment. So here I'm going to show you some data. Here we use Western blot analysis to show that in all of these cell lines, interleukin 24 phosphorylates eIF2 alpha at serine 15. And below we have our cell proliferation and apoptotic assays. So all of the cell proliferation assays indicated that interleukin 24 was indeed killing these cancer cells. The same results were seen with the annexin V apoptosis assay. So interleukin 24 was indeed inducing apoptosis in these cancer cells. So next, we wanted to directly determine whether eIF2 alpha was necessary for interleukin 24's killing effect. So here, So here we use Western blot analysis to show that interleukin 24, well, before we show that interleukin 24 does indeed phosphorylate eIF2 alpha. Next, we wanted to employ eIF2 alpha mutant cells. Okay, so we treated, so here basically we were able to get cancer cells from our collaborator at Harvard, and these cancer cells contained a mutant form of eIF2 alpha. So these cancer cells were genetically engineered so that the eIF2 alpha, this phosphorylation site, would be mutated. So instead of a serine at specific amino acid 51, the serine was mutated to an alanine. So in effect, what would occur is that interleukin 24 or any other kinases would not be able to phosphorylate the specific eIF2 alpha. So basically what we hypothesized was that if this specific eIF2 alpha could not be phosphorylated, then we would not have a decrease in global protein synthesis. So if interleukin-24 is unable to cause the phosphorylation of the mutant EIF2 alpha, then translation initiation would still occur, and we would get protein synthesis, and the cells will continue living. So here I have some data. We treated both mutant cell lines and wild-type cell lines with increasing concentrations of interleukin-24. So as expected, the wild-type cells showed killing effects in response to interleukin-24. So as we increased the concentration, the cells died. We can also see that in our apoptosis assay. The cells were increased in their apoptotic effect in response to increasing concentrations of interleukin-24. When we looked at the mutant cells that did not have the phosphorylatable form of EIF2 alpha, there was no significant change in apoptosis or cell viability. So we also performed a Western blot to show that with increasing concentrations of interleukin-24, we see increasing levels of EIF2 alpha phosphorylation in the wild type and no phosphorylation with the mutant cells, confirming that our mutant worked and the inhibition of interleukin-24's killing effect was due to the fact that EIF2 alpha could not be phosphorylated. So overall, we show that interleukin-24 phosphorylates EIF2 alpha to induce apoptosis in cancer cells, and it does this by depleting the ternary complex, which cannot initiate translation and create new proteins for the cancer cells. Currently, we are working on another arm of translation initiation in which interleukin-24 may also decrease the EIF4 complex here, which is required for mRNA binding at the 5' untranslated region for protein synthesis. Many oncogenic proteins that promote tumor growth contain long 5' untranslated regions. So if the EIF4F complex is not available, maybe due to interleukin-24, these mRNAs cannot be translated to protein, which will decrease cancer cell survival signals, leading to cell death. So here we are. If both of these arms are affected by interleukin-24 overexpression, that will inhibit translation and thus allow the cells to undergo apoptosis and cause cancer cell death. So in terms of our continuing and future studies, we are looking at how interleukin-24 expression affects the different pieces of the EIF4F complex. We want to find out also how does interleukin-24 affect mRNA stabilization for protein synthesis. So this is one of the continuing projects that we are currently working on. We want to also verify if interleukin-24 does downregulate oncogenic mRNAs. We are also looking at additional mediators of interleukin-24-induced apoptosis. We recently found that protein kinase A, also called PKA, is a major cell regulator that plays a role in regulating interleukin-24's mediated extrinsic apoptosis, which works through death receptors rather than internal signals that tell cells to die. We are exploring the role of PKA in various different pathways that interleukin-24 normally activates to kill cancer cells. So we're trying to find out if PKA is a major regulator in interleukin-24-mediated cell death. We also want to know how interleukin-24 mediates its own expression. So what transcription factors are involved in expressing the interleukin-24 gene, and what proteins are involved in stabilizing interleukin-24's mRNA. Also, sigma-1 receptor is involved. So as I mentioned earlier, interleukin-24 will bind to sigma-1 receptor to potentiate ER stress in the cancer cell to cause apoptosis. So we are interested to know if sigma-1 has any additional roles in respect to interleukin-24 expression of mRNA. So overall, in basic research, through our studies of the signaling pathways and respective proteins, what we hope to achieve is to find new targets for anti-cancer therapeutics. If we understand the proteins that interleukin-24 targets to kill cancer cells, we could possibly create drugs from those binding domains to treat cancer patients with. Also, by understanding these pathways, we can develop combination therapies and personalized therapies with interleukin-24 that will kill cancer cells in a more effective and synergistic manner. So there have already actually been studies showing that in combination with interleukin-24, well-known therapies such as Herceptin and cisplatin, they actually work better with interleukin-24 treatment. We also want to extend the positive results seen with melanoma patients in clinical trials to patients with other cancer cell types. And as we have shown, interleukin-24 targets the protein translation, which is an essential process to all cancer cells. So this would be a key in developing interleukin-24 as a potent anti-cancer therapeutic against multiple cancer cell types, not only melanoma. So overall, to conclude, I would like to thank, of course, Dr. Moira Sauane. She's an amazing mentor. I've been working with her for the past 2 years, and she always has really great ideas, and she inspires me every day to be a great scientist. I definitely want to thank my current and former lab members, Zwelin, also known as Sean, Anna, Hilal, Marifer, Carlos, and Jordan. Special thanks to our collaborators, Dr. Akhtas from Harvard Medical School and Dr. Redenti, and fellow PhD student Giessen, who are our lab neighbors at Lehman College. And of course, none of this research could have been done without funding from NIH and the National Cancer Institute. So thank you everyone for signing in today to listen to my presentation. I'm happy to answer any questions, and I welcome you to connect with me on Twitter or LinkedIn if you would like to learn more about what we do at Lehman College. Thank you.